Hey class, here is my presentation, the first one on the leucine zipper and the prompts that we were asked to talk about. So I have a couple slides here. This first slide, this is going to be that picture of that process of the double helix and the leucine zipper that we have to discuss. So a little about this. So this is a leucine zipper domain, and it's comprised of two long intertwined alpha helixes with hydrophobic side chains extended out from each helix into the space shared between them. The hydrophobic side chains are the leucines, given this domain its name, of course. Note, I don't know if you can see it from the picture, but that there are tight packing between the leucine zipper helixes. This makes the domain very stable. The extension of the two leucine zipper helixes stagger the DNA major groove, and you can see the major and the minor groove there. Side chains from both helixes extend into the groove to contact the DNA bases. The specific interactions between side chains and bases are, of course, the hydrogen bonds. So we were first asked one possible residue that could replace this leucine. I found that isoleucine would be a good example of this. This is a residue that could replace leucine because it is equally as hydrophobic with the same chain length and molecular mass. It also has these similar properties. The one I didn't mention was the neutral charge and the alpha thetic property, which just means that the carbon atoms form open chains and not aromatic rings. So then we were asked the opposite of the first prompt, or the first question, I'm sorry, as to what could destabilize the core of leucine. So I think the takeaway for this is any hydrophobic residue can destabilize the core by breaking the hydrophobic interactions that are present. But an example of this could be lysine. And here's just a chemical property of lysine. Lysine could do this because unlike isoleucine, it is hydrophilic and it has a positive charge, so it's the opposite of the isoleucine, so that could destabilize the leucine. The next part of this was propose a different amino acid pair whose side chains could interact in a similar manner to lysine and glutamate. So from researching this prompt, I realized that any side chains of any pair consisting of one acid and one base amino acid will interact like lysine and glutamate. So there are multiple examples. Two of them could be aspartate and arginine, or aspartate and histidine. But like I said, they're both the same, but for this one, it has one basic chain, it has one acidic chain, and all polar and hydrophilic, so it does act like the leucine.